Hi there again. Hopefully you had a good break. We're going to resume our mini lecture series here in Unit 4 Animal Nutrition, and in this section we will talk about digestive systems. But before we get to that, just want to review real quickly the 8 basic feeds that we're going to be talking about, or feed classes. First is, of course, dry roughage, which is like your hays. Of course, you can't talk about feeding without pasture. Silages is number 3 here, and that would include anything wet forage, whether it's corn silage, alfalfa silage that's chopped wet and stored, fermented, wheatlage, any of those kind of things. Energy feeds, which is of course corn and other cereal grains like that. Protein feeds like soybean meal. Mineral feeds, which we talked about, as well as vitamins and additives. And you can kind of see a little bit the Rumensin logo here, which is a ionophore that helps with digestibility. So let's talk about digestion in the next few slides here. And digestion is both mechanical and chemical in nature. You have to have both things occurring effectively and efficiently in order for us to break down the feedstuffs that we feed our animals in order for them to produce in a manner that we expect. And so those feed particle sizes have to be reduced either to basically to its smallest form, whether that's amino acid, whether that's a glucose molecule, whether that's a mineral ion. Let's talk specifically first about the monogastric. So like the pig, the dog, the chicken, or the horse, good examples of these. And we're going to start up in the mouth, and we have both, in a monogastric, both chemical and mechanical digestion occurring. Of course, we have to think about the mechanical part of the chewing. We have to be able to bite off and chew our foodstuffs into smaller particles in order to be able to swallow what we call a food bolus down the esophagus. But in the monogastric, you also have salivary glands, of course, that not only emit a little bit of mucus to soften the food, but there's also pepsins, enzymes, and a little bit of acids in there in order to already begin that chemical digestion process. It is different from the ruminant, which we'll talk about later. The esophagus is just very simply a delivery tube. Just think of a garden hose. There's really nothing really important about the esophagus except the fact that it's there. There is a valve at the lower end of the esophagus, and that flapper valve is supposed to keep acids and foodstuffs that are in the stomach from coming back up, or any of those acids from splashing up and eroding the esophageal lining. The stomach then is, of course, the primary area of feeder particle reduction size. That can occur whether it's mechanical or chemical, because the stomach will churn and mix feedstuffs in order to help with breaking things down chemically. And of course, then we have chemicals in the stomach that will cause a lot of chemical reactions to help break down feedstuffs. In the upper part of the stomach, you'll see alkaline substances like mucus emitted into the stomach to help lubricate the feed. And the lower part of the stomach is where a lot of those acids come into play. Moving on, then, of course, as feedstuffs exit the stomach, they're going to enter the small intestine. And the first part of the small intestine is the duodenum. And that will not only help in splitting molecules in that upper part of the duodenum, but also you begin then the feed absorption process, or the particle absorption process. And that continues into the middle section and the lower sections of the small intestine, the jejunum and the ileum. The large intestine then hooks onto the end of the small intestine and will contain the cecum, the colon, and a few other structures on occasion. And its primary function is to resorb water, is to suck water back out of that feed stuff, help to dry it up a little bit, and then form the indigestible fecal material that will be expelled from the body. Here's a good diagram of a pig. Actually, our digestive tract and the pig's digestive tract are almost identical. They're very interchangeable. Our stomach sizes are almost exactly the same. So it's important, and we'll do a lab experiment later on in the semester and do some dissection, and you get the chance to study these things. Here's the chicken, and essentially a chicken doesn't have a true functioning stomach. They, of course, have the crop and the proventriculus and the gizzard, which all have a little bit different function, but do some of the similar things that the stomach does in for example, the pig. And you see theirs is a little bit different in terms of the length of the small intestine and where some of the other structures are located. Consequently, here's the horse, and the horse is unique. The horse is classified as a monogastric, but actually functions much like a ruminant. A horse's stomach is relatively small and not that much bigger than a pig or yours and mine, to be honest with you. The small intestine is also relatively much shorter. The unique part of the horse, and I can't stress this enough because it will be in your assessments, the unique part of the horse is the cecum. The cecum is a great big fermentation vat. So all of that pasture grass or hay that your horse eats, most all of that passes all the way down through the tract all the way to the cecum, where it will ferment and break down from that point in time. And so basically 60% of the digestion in the horse occurs in what we call the hind gut, which will contain the small colon, cecum, and large colon area. So that's important to recognize that the horse is quite a bit different. So again, back to our discussion of monogastrics. That stuff is going to begin in the mouth, and you're going to have a number of different things occur here. I'm going to click through a couple of these slides quickly because I don't want to exceed my time in this section. But there's two methods of absorption I want to talk about. Passive absorption, where the molecules that are in the small intestine are just going to pass from high concentrations to low through the act of diffusion. The other method by which feedstuffs can be, and molecules and amino acids and so forth, can be drawn into the lymph system, and that is active transport, where the interior of the small intestine is covered by millions of little finger-like hair structures, and those are called villi. They will actually reach out, so to speak, and capture amino acid and pull it into the lymph system and pull it out so that it can be transported through the body via blood. Of course, liver function is important in here because it's going to help metabolize a lot of different feed particles into the bloodstream. It's your filter or detoxification chamber. In order, if there's any of those kind of toxic substances, it'll capture those and get rid of them for you. So let's transition and talk about ruminants, and I think this will wrap up this second mini lecture series. I didn't mention anything about the mouth here in ruminants, but ruminants don't have upper incisors, and they also don't have salivary glands that emit chemicals. It's only mucus. So the ruminant mouth is offset a little bit. The teeth are offset, the jaw is offset, so it serves more of a grinding rather than a chopping function up in the mouth. The esophagus is the same, of course. But then we're going to dump into a very different 4-compartment stomach system. The rumen is the first part that we'll talk about, and it takes up essentially the whole left side of the cow between the shoulder and the hip. And it is a great big fermentation vat filled with millions of bacteria and protozoa that help to break down a lot of the forages that ruminants will consume. That rumen environment should maintain a pH of around 6.2 to 6.8. And that is really important because if it gets too much more acidic, some of those microbes will be killed. And then it's also lined with thousands of papillae, or finger-like projections, that will help capture volatile fatty acids, potentially even amino acids that are made in the stomach, in the rumen, and transport them throughout the body. Feedstuffs then also would move through the reticulum, which is known as the honeycomb. You'll see some photos of this as we go along here. And this provides the initial mixing action of the rumen that will mix about 3 times a minute. It also captures any foreign substances, rocks, maybe a nail, consequently, that might accidentally get into feeds. The omasum is next, and in an adult cow, the omasum is roughly the size of a basketball, and it's called many plies, or leaves of the Bible, and it will have several 100 folds of skin with barbs on it, and all it does is basically grind feed stuff. It's a relatively dry environment, so there's not a lot of digestive responsibility here. It's primarily feed particle size reduction. And then from there, from the omasum, feedstuffs pass into the abomasum, which is the true stomach of the ruminant, and functions identically to the monogastric stomach that way. Ruminants are always going to go through the process of what we call rumination. So they're going to actually regurgitate boluses of feed up from the rumen and rechew them and resalivate them and then reswallow them. That's important for not only reducing more feed particle size, but also maintaining a healthy rumen environment and a healthy rumen pH. As a consequence, of course, of fermentation, we have gases that we have to get rid of. Otherwise, we have some bad things that could occur. But those gases are belched out through an eructation process. So cows will always be burping, getting rid of that extra methane gas that's going to be a byproduct of that fermentation. So here's again another diagram, a cross section of a ruminant, and you see here from the head on the what would be the left side of the steer here. He's got a rumen that takes up again that whole left side. Same thing on the top here on the back side of that photo. And then the omasum sits here, the abomasum sits at the bottom. It's not showing you the reticulum, which is kind of right at the front, right next to the heart, consequently. Here's some photos of different stomach structure, the 4-compartment stomach structure. You can see the reticulum here on the top photo, the rumen here on the bottom left, that reticulum, see that honeycomb structure, the many folds or many plies of the omasum, and the abomasum is primarily smooth tissue. A specialized structure in ruminants is what we call the esophageal groove that really only functions in a young ruminant and is primarily meant whenever a calf is suckling or a young is suckling to divert that milk product directly to the abomasum because we don't need milk fermenting in the ruminant. You guys have all smelled fermented milk probably. You wouldn't want that fermenting in your stomach. So that's the function of that thing. Digestion in ruminants is high function and highly correlated to fermentation. And microbes are a big part of that, like I mentioned earlier, and the production of volatile fatty acids, which we will discuss in class in much more detail. We mentioned eructation, and if this action fails, we get a health condition called bloat. And we can talk about that in more detail in class, but that can be fatal to ruminants. And so it's important that ruminants are ruminating appropriately and getting rid of that gas. Rumens aren't really completely functional until about 2 months of age in sheep and about 3 or 4 months of age in cattle. So it's not really that beneficial to feed them high forage diets younger than that. So most of their diets should be made up primarily of grains. A little bit more on digestion here, and I'm not going to spend much more time on this. You guys can pause this slide or recheck the notes here because I want to wrap this section up on digestion of ruminants. A little bit here of a diagram on size of what the part of the digestive tract would be able to contain. Here's some further diagrams that shows you the complexity of energy pathways in the ruminant, where energy goes. Same thing with protein pathways. So we're going to pause here and wrap up mini lecture 2. Make sure you've taken good notes, written down some questions for the muddiest points, and I will look forward to discussing these with you in class.